It's really exciting. As you said, my name is Breanna Whitley, and I started this project as an undergrad. So it's really amazing to be here at the culmination of this project and to have you all with me today and to be in person, but also a little bit on Zoom too. So thanks again for being here. And let's get started talking about my project, which is the phylogenetic, morphometric, and biogeographic investigations of Trema micrantha, which is in the Cannabaceae family, also known as the cannabis family. So many of you are probably familiar with the cannabis family. You might know of cannabis, or you may know of humulus, which is used as hops for brewing. But you may not know about this really interesting group, which is Trema. This group is a pan-tropical genus of pioneer trees. So these are often the first plants that are colonizing disturbed areas. And they are important components of successional vegetation following those disturbances, whether that be landslides, maybe reclamation after agricultural use, etc. And this is an economically and ecologically important group. So obviously ecologically important because they are a pioneer species, and economically important because some lineages, such as Trema orientalis, which is a paleotropical or old world lineage, demonstrate different pharmacological properties. So it's been found to have antiplasmodial activity, which can help treat malaria, and also anti-cancer activity, which is really interesting. And traditionally, these lineages have also been used to treat diabetes, various inflammatory diseases, and muscular pain by indigenous people. And in the neotropics, Trema micrantha is often used in habitat restoration projects to reclaim agricultural lands. And also it's been an important resource for paper production for various peoples in Mexico, such as the Otami. And you can see some of the bark paper designs that these people create using Trema micrantha bark. So that's pretty interesting. And when we think about this group taxonomically, it's been pretty problematic both in the neotropics and in the paleotropics. So there are currently about 20 recognized species and 2 accepted varieties, but there's 66 synonyms within this genus and about 10 ambiguous names. So you can kind of see where this problem is really concentrated. And this is likely due to the fact that Trema has never been monographed in its entirety, and that has led species concepts to vary geographically based on local floras. So focusing now a little bit on the neotropics, there's typically 4 species that are recognized. So first we have Trema cubensis, which is distributed in the Dominican Republic, Haiti, and also in Cuba. Then we have Trema lamarckiana, which is a Caribbean distributed species, also found in Florida. We have Trema domingensis, which is found in Central America as well as South America. And then we get to the group that I'm really interested in, which is Trema micrantha. And as you can see, this is the most broadly distributed of those neotropical lineages. So this is what those lineages look like. So we have Trema lamarckiana, which typically has smaller leaves. And then we have Trema cubensis, and this has a montane distribution. It has nice little tiny kind of ovate leaves, and it typically has very round teeth along the margin. And then we have Trema domingensis, and this group is the only species that has an entire leaf margin, which means that there's no teeth along the edge of the leaf. And then we get into Trema micrantha, and as you can see, just based on these 3 specimens, this species is pretty morphologically variable. And it's distinct from Trema domingensis in having teeth along the margin. So as I mentioned, this group is especially problematic, and of those 66 synonyms that I mentioned previously, 51 of those are concentrated in Trema micrantha. So authors sometimes characterize this as a highly variable species, or sometimes they might divide it into different varieties or distinct species, often based on leaf shape and trichome morphology. And trichome just means hairs. So there's hairs on the abaxial surface on the leaf that are often important for differentiating this group. The flowers are pretty uniform, so that's often not used to distinguish species. So along with being morphologically variable, this group is also not monophyletic. And that just means that all these lineages that we've identified that are described as Trema micrantha, they don't share a common ancestor. So if we look at this just simple cladogram here based on 5 DNA loci from a previous publication, we can see Trema micrantha A and Trema micrantha C. And these are distinct from the Bs over here. So B1, B2, and B3. And Trema micrantha C is unique because it has a montane distribution and it also has a distinct leaf morphology. And then Trema micrantha A is also distinct. But then when we get into the Bs, which I'll be focused on today, they're a lot more complicated, and that's why I'm focused on that group in particular. And they're not monophyletic, they're morphologically variable, and as you saw in the previous slides, they're also very broadly distributed. And in that broad distribution, they occupy lots of different habitats. So these are all Trema micrantha. So you can see Trema micrantha growing in Florida along like a power cut. Then we see Trema micrantha growing in a more arid environment in Brazil. And then Trema micrantha growing in what looks to be a secondary forest in Mexico. So hopefully you can kind of see the problem that we're trying to address. And that problem is that this group is polyphyletic, so not all the lineages share a common ancestor. They're morphologically variable and very broadly distributed. So when we think about this, we see that this is likely not an accurately described species that needs a little bit more work. So the question that I'm trying to address is, are these morphologically variable and phylogenetically divergent lineages of Trema micrantha, can they be diagnosed as distinct species? And again, I'll be focused on this B group for the remainder of the talk. So to answer that question, I have a multifaceted approach that includes phylogenetics, so looking at the evolutionary relationships of this group, morphometrics to see if these are morphologically distinguishable, and then also biogeography, so looking at the distributions of the various lineages. And in using this approach, we can then satisfy or evaluate different species concepts. So with the phylogenetics, we can look at the phylogenetic species concept. Are there lineages within this group that are monophyletic? We can also look at the morphological species concept. Are these morphologically diagnosable? And then we can look at the ecological species concept and determine if these lineages are occupying different ecological niches. All right, so the first question relates to the phylogenetic portion of my thesis. And that question is, how many lineages are actually present within Trema micrantha, specifically the B group? And is there strong support for those lineages? So to answer this question, we build a phylogeny to understand the evolutionary relationships and identify clades. And in systematics, the phylogenies that we're building, we're looking at molecular data. So to get molecular data, which means DNA data, we need to do some sequencing of that DNA. So for my sequencing, I have two different methods, the first being Sanger sequencing. And we looked at 5 DNA loci, so 3 of those being from the plastid genome, 2 of those being from the nuclear genome. And this results in about 4,000 base pairs. Then I've also looked specifically at the external transcribe spacer, and this is a nuclear locus, so we can begin to ask questions about hybridization between lineages, and I'll talk more about that. And this is about 300 base pairs. And then we've also employed high throughput sequencing, which gives us a lot more data, and this gives us genomic data. So with the high throughput sequencing, we've obtained the nuclear ribosomal cistron, which is about 6,000 base pairs, and then also the plastid genome or plastome, and that's about 150,000 base pairs. And this is just the layout of the nuclear ribosomal cistron. This is tandemly repeated in the nuclear genome in this order. So we have the external transcribe spacer, 18S ribosome, and then the internal transcribe spacer 1, 5.8S ribosome, ITS2, a 26S ribosome, and then the intergenic spacer. And then this is kind of the layout of the plastome. So before I started this project, the sample size for the Neotropical Trema group was about 24 specimens, and that was published by Garwood et al. In 2018. And again, Sanger sequencing is nice because it targets specific loci, and it's relatively inexpensive and only takes maybe a couple weeks to extract the DNA and send off your data and then sequence and analyze that data. And this allows us to have a broader taxon sampling. So specifically, as a result of this project, now we have a sample size of about 207 for the neotropical lineages. And while this allows us to have a broader taxon sampling, because we have less character sampling, so if we think about those 4,000 base pairs that I mentioned, sometimes this may lead to less phylogenetic resolution, but not always. And then again, as I mentioned, I've also looked at the external transcribed spacer. And this was a way for us to increase sample size and sequence specimens that weren't sampled in various data sets. So once we identified clades based on the other data sets, then we can look at the external transcribed spacer, which is variable among species, and we can see we can assign specimens to different clades. And then, as I mentioned, we can also identify hybrids because this has biparental inheritance, meaning that has signal from the mother haplotype and the father haplotype. And this is where we have the broadest taxon sampling. So you can see N equals 489, and that's for Trema micrantha. But we have the least amount of phylogenetic resolution because we have the least amount of character sampling. And then for the high throughput sequencing, our sample size for Trema, the neotropical group, is 62. And in this sequencing, you get lots and lots of DNA, about millions and millions of reads from high copy regions of the genome. And this is where we have less taxon sampling, so smaller sample size, but we have improved phylogenetic resolution because we have a lot more character sampling. So for the sequencing methods, first you start by looking at your specimen and collecting tissues from that specimen, extract the DNA, and then since we're targeting a specific loci with Sanger sequencing, we need to amplify that loci and get lots and lots of copies, which is shown in number 3 here. And then you can load your sequencing plate, send it off for sequencing. And then as a result, you get your data, which looks like this in number 6. And you can see all the different nucleotide calls at their various positions. And that's the kind of data we get for the ETS sequence data as well. And here we can begin to, as I said, assign different specimens to clades based on their the ETS data. And this region has biparental inheritance. So we can see that lineage 1 at a base position 2 has a T. And then for lineage 2, we can see at base position 2, it has a C. And then when we look at this middle one here, we can see at base position 2, we have both a C and a T, which likely means that this could be a putative hybrid lineage because we're seeing signal from both of the parents. And in my data set, we've detected 73 hybrids within Trema micrantha group B. So that's only about 25% of our samples. And those were removed from the phylogenetic analyses because they're hybrids, they can pop up in different clades and it can make things kind of complicated. So for the high throughput sequencing, we do the same methods where we collect tissue samples, extract the DNA, but then we send it off and they do all the complicated library prep and sequencing. And what we get back is, as I mentioned, we get reads from all the high copy portions of plants' genomes. And if you aren't aware, plants have 3 different genomes. So we have the plastome, which is shown in green here, the nuclear genome in purple, and then also the mitochondrial genome. So when we get back this data, we need to filter that data and retrieve the parts that we're interested in. So for me, that's the plastome and the nuclear ribosomal cistron. So you go through various filtering methods, and then you build your plastome or your nuclear ribosomal cistron with different reference assemblies, de novo assemblies, do a lot of data checks, and annotate the genes and go through various processes. And this can take from months to years sometimes. So then once we have that data, then we can build the phylogenies. And I use maximum likelihood with IQ tree and also Bayesian inference with Mr. Bayes. And I use the Big Dog cluster, which is an amazing resource available here on campus. I did that for all my data sets. And then for the ETS locus, because I was interested in assigning specimens to clades, I used a UPGMA, which is essentially like a distance analysis to identify genotype clusters. And I'll talk about that more. And before I get into the results, for those of you that aren't familiar with systematics or taxonomy and the process of describing species, you may be wondering, as I present my results, well, how did you decide which clades are which? And so I'd like to just kind of share that information with you real quick. So how do we designate clades whenever we're pursuing these systematic pursuits? Well, first we want to observe the morphological diversity of the organism that we're working with. And so for some people that may be going out in the field and making collections, but for us that meant making trips to Missouri Botanical Gardens and looking at thousands of Trema micrantha specimens, and also requesting loans from other institutions such as New York and other herbaria as well. And when we're looking at this diversity, we're looking for different morphological groups maybe. So we might see specimens that are very similar. We might want to sequence those. And then we also see specimens that might be really different that we haven't sampled yet. So when we're looking at this morphological diversity, we want to sample across that morphological breadth, but also the geographic breadth of this group. As I've mentioned, this is a very broadly distributed group, so we want to make sure that we have adequate geographic sampling as well as morphological sampling. And once we've done this, then we can sequence those specimens we can sequence those specimens that might represent different morphological groups. And after sequencing, we build the phylogeny, and then we want to look for the least inclusive phylogenetic group. So we want to look at clades. So for example, you can see my cursor here. So we might have, this might be a clade here, but then we might look within this clade and see that there's a subclade. So we want to find the least inclusive group of these organisms. And then when we find that group, we're going to go back and again, look at that morphological diversity. Is there some morphological signal that represents this lineage? Is it from a unique geographic distribution? And we're going to go through this process again. So this is an iterative process where we're constantly thinking about the relationships of this organism and also their morphology and their geographic distribution. All right, so now time for some results. So this is the 5-gene phylogeny. And what we can see is that we've identified 5 clades within Trema micrantha B. And these are represented by the different colors. So we have B1b, B1a, B2, B4, and B3. And these are just ways for us to kind of keep these clades straight. And we see that these clades are monophyletic. So that means that all of the descendants share a common ancestor. And we see the monophyly also of some lineages I mentioned earlier, which is Trema micrantha A and Trema micrantha C. So we always see that these are well supported as lineages or species. But I also like to mention, you might be able to see this little clade here, and that's Trema micrantha D. And with this increased taxon sampling, we've also recovered another lineage that's pretty interesting. It's only found so far in Cusco, Peru, and it has unique morphology. I think they're really cool looking. So that'll be interesting for someone else to work out. So then here's the plastome phylogeny. And again, we see monophyly of those B groups. But this time you might notice that B1b, that orange lineage, is in a clade with B1a. So it's not necessarily monophyletic. And we still see the strong support of A, C, and D. And then for the nuclear ribosomal cistron, we see monophyly of several of those B groups. And this time B1b is now in a clade with B3, but it is monophyletic. So it's nested within that B3 group. And we still see the monophyly of A, C, and D. Then for the ETS data, I did a UPGMA analysis. And this is just essentially, as I said, like a distance analysis. And with this increased taxon sampling, so remember that for ETS, we had about 489 specimens that we sampled. With that increased sampling, we've also recovered this B5 lineage that you can see in the blackish color. That's sister to B3 in this group, in this analysis. So that's pretty interesting too. So we have another clade that we should think about. All right, so now we've done the phylogenetics, so let's think about the morphology. And the question that I'm addressing for the morphology is, do the phylogenetically divergent lineages within Trema micrantha B have distinct leaf shape that can make them morphologically diagnosable? So to answer this question, we need to really evaluate leaf-shaped characters or morphological characters. So to do that, I took some various measurements of leaves. So blade length, blade width, distance from the base of the blade to the widest point, basal indentation, which is the measurement from the base of the leaf to the insertion point of the petiole, apex length, apex width, petiole length, and teeth density. These were all taken in millimeters using a ruler or sometimes on images using image J. And we took measurements of 3 mature leaves per specimen. So the largest leaf, the smallest leaf, and then an intermediate one. Then from there, I calculated the mean values of each of those measurements and included those in a principal component analysis. So with this principal component analysis, we can display the leaf shape variance and see if these lineages are occupying different morphological space. And we can also reduce the dimensionality of the data, which means that we can determine which characters contribute the most to the leaf shape variance, and then reduce the number of variables that we use in a one-way ANOVA. So with those one-way ANOVAs, we can look at character by character if they're statistically significantly different between lineages. So here's some results. So first thing I'd like to mention is that based on the eigenvectors, we can see that blade length, blade width, distance from the base of the blade to the widest point, apex length and apex width are the most important variables, contributing more than 20% to the variance that we see in the principal component analysis. Now this might look kind of messy, but as we move forward and discuss the results a little bit more, hopefully you can see the patterns that I see as well. So we'll just wait to get there until just a moment. And then here's the results from the one-way ANOVAs of the various characters. So again, the most important characters that contributed to that leaf shape variance. And I'm going to talk about these in detail as we move forward. So just keep that in mind. All right, so then the last portion of my thesis was the biogeographic questions that we have. And so that question is, what ecological niche does each of these lineages occupy? And to answer that question, we need to map lineage distributions and then model their ecological niches. And this is how we do that. So on each herbarium specimen, there's locality information. Sometimes if you're lucky, it's geographic coordinates. And so that's what we use for our occurrence data. And then when we're building these models, we need occurrence data. We also need environmental data. So we might use things like precipitation or temperature. And then from there, you can build a correlative model where you have occurrences, your environmental data, and then as a result, you get some habitat suitability. So for the environmental variables, I use the 19 bioclimatic variables from Chelsea, and these are all related to either temperature, precipitation. And then I also use soil variables, and those are the 12 variables from ISRIC, and both data sets were at 1 kilometer squared resolution. And then I removed correlated variables greater than 0.8. There were no soil variables that were correlated, and these were the resulting bioclimatic variables after removing correlated variables. And then because we're working with historical specimen data and we're building models, we want to make sure that we don't have biased data. So we need to reduce sampling and environmental biases. And I did that by calculating the climate heterogeneity and then filtering that occurrence data based on that heterogeneity. So what that means is if we had a cluster of occurrence data, let's say in this dark blue area where the climate is pretty homogeneous, so it's not very different. So then it would filter that occurrence data more liberally. And then if we were, if we had a bunch of occurrence data in these reddish to green areas, that's more heterogeneous. So then it would filter that data less. And then models were tuned and parameterized in SDM Toolbox. And the ecological niche models were created with Maxent software, and model performance was evaluated using the emission error rate, the OER, and AUC, which is the area under the curve. And don't need to worry about those definitions much, but what this tells us with the AUC is it tells us false positives. So how much unsuitable or uncolonized habitat, so habitat where we don't have occurrence data, is estimated as suitable. And a perfect score for AUC is 1. So we want scores that are close to 1. And then for the emission error rate, this is our indication of false negatives. So how much suitable habitat where we have known occurrences is estimated as unsuitable. And we want that score to be close to 0. So now for some results again. So this is the distribution of the lineages that I mentioned earlier in the phylogenetic analyses. And we can see that there is some overlap, some geographic overlap between these lineages, but there is also some that are geographically distinct. And here are the ecological niche models. And if we look at the AUC scores and the OER scores, we can see that these are pretty good models, some of them being really good. So I would give these all a thumbs up. But I will mention that something that's interesting and I think somewhat expected is that our more broadly distributed lineages have poorer scores or lower scores. And like I said, I think that's to be expected, but these are good models. Okay, so now we've gone through all of the methods, and now we're to the point where we've identified our clades, we've done the analyses, and now we can define niches and morphological characters for each of these lineages. So just as a reminder, in our 5-gene phylogeny, we have monophyly of these 5 lineages. And when we look at the plastome and the nuclear ribosomal data, we see that B1b, that orange lineage, is not reciprocally monophyletic. So it's switching positions in these 2 data sets, and that's kind of to be expected, and I can talk about that more. And there's more complicated relationships that are worth noting that we'll likely discuss in my defense, but we don't have enough time to talk about it today. All right, so again, I'd like to kind of share with you some of the results that we found. And here's representatives of B1A and B1B. So these are what I call the B1s. And we can see just looking at these representatives that these are pretty, to me, they look pretty different. They look morphologically distinct. We see B1A has a more oblong or elliptic leaf, while B1B has a more ovate leaf with a nice cordate or harp-shaped leaf base. And they also have different trichome morphology, which I'll talk about. Whoops. And if we look at the PCA that I showed earlier, we can see that B1b, this orange lineage, is somewhat morphologically distinct from B1A in this analysis. So we can see that they're kind of overlapping, but they are occupying some different morphological space. And we can see that very clearly that they are occupying different morphological space from B2 and B4. So that's the green and purple lineages. And then I'll talk more about B3, the blue one, later. And when we look at the one-way ANOVA, which looks at the individual characters, we can see that blade length and distance from the base of the blade to the widest point was significantly different between these, what we call sister taxa. And if we look at those a little bit closer, we can see that blade length was significantly different. And if we look at these box plots and you see, if I can find my cursor. Well, anyways, if you look at the box plot for the orange one, you can see that the mean value, that's the black bar in the box plot, is lower than it is for B1A. And if we look at these representatives here, we can see that B1A tends to have longer leaves and B1B tends to have shorter leaves. So this makes sense. And then when we look at distance from the base of the blade to the widest point, and this is kind of our proxy for leaf shape. So if you have a distance from the base of the blade to the widest point that's smaller, then that tends to show that this leaf is more ovate in shape. And so if we look at B1b, we see that has a smaller mean value, and we see that B1A has a higher mean value. And this is the same pattern that we see when we observe these specimens. So you can see B1A is more elliptic in shape, B1B is more ovate in shape. And when we look at trichome morphology, we can see that these are pretty distinct too. So again, these are hairs on the abaxial surface of the leaf. And for B1A, they have shorter, more sparse, oppressed hairs, while B1B is very hairy, has tomentose hairs covering the entire bottom surface of the leaf, and they're very fuzzy. And then when we look at the ecological niche models, we can see that, again, these lineages are somewhat overlapping in their distribution, but they are also somewhat distinct. And when we look at the variables, we see that they share the same first variable, which is temperature seasonality. But when we think about this a little bit more, we think about these habitats where they're occurring, we can see that B1A is distributed in humid tropical savanna and tropical monsoon climates. And it's typically found in high evergreen forests. And this is a species that has a relatively moderate to high elevation. So the mean elevation of the specimens that we sampled is about 1,027 meters. And about 84% of the occurrence points that we have for this group have elevations over 500 meters, and 56% of the occurrence data that we have occurs over 1,000 meters. So this is, again, a moderate to high elevation lineage. And then we look at B1B. This is a lowland species where the mean elevation is about 273 meters. And it's found in very seasonably very dry areas and deciduous forests. So if we think about the Yucatan, that's a generally pretty dry area. So based on this evidence, we can see that B1A and B1B, they fit the phylogenetic species concept as monophyletic groups, and they also fit the morphological species concept. And while maybe the ecological niche models didn't inform us that they might be ecologically distinct, when we think about the distribution of their elevational preferences, I believe that these also satisfy the ecological species concept, and they can fall nicely within this Venn diagram in the center. Then we have B2 and B4, and so B2 is the lineage that's distributed throughout South America and Central America, and B4 has a more limited distribution. But just looking at these specimens, to me, they don't look that much different. And when we look at the principal component analysis, we kind of see that same pattern where the purple lineage representing B4 and B2, the green lineage, are overlapping in this analysis. But I would like to mention that we have a relatively small sample size of B4. So it's also kind of hard just with that limited data to parse out this group. So we see that they're maybe not morphologically distinct. And in the one-way ANOVAs, they were only significantly different for distance of the base of the blade to the widest point. And I'm not sure that I really observed that same pattern as well. So there's more work to be done to distinguish these two from each other. But when we look at those B1s that we just talked about and B2, we can see that they're occupying different morphological space, as I mentioned earlier. And they were significantly different in 4 of the 5 characters that we looked at within ANOVA. So the ones I'd like to highlight specifically are apex length and apex width. And we see that B2 tends to have nice long apices, a more attenuate apice or apex, sorry, while the B1s have a smaller apex. And now looking at their ecological niches, we can see that B2 is more of a generalist species where it's kind of occurring everywhere, but it's also a lowland species, and it's often occupying gallery forests, semi-deciduous seasonal forests as well. And again, this is a very kind of generalist species. But then when we look at B4, we can see that it has a more limited distribution, and it's actually occurring on the eastern foothills of the Andes along the Cordillera Real throughout Bolivia and Argentina. And this is an area that's very humid, and it has very distinct precipitation seasonality patterns. So this is pretty interesting. And now based on that information, I think that these lineages are satisfying both the ecological species concept and the phylogenetic species concept. And based on the morphological data, I can't really say for sure based on that data that they're both morphologically distinct, but just looking at the difference between B2 and the B1s, I think that B2 could also fall into the middle of this Venn diagram where it's satisfying all three species concepts. And then that B3 lineage, this group, you can see that according to leaf shape, it looks kind of similar to the other lineages that I've shown so far. But you might notice that these are very grayish to white on the abaxial surface of the leaf. And that's because they have very distinct trichome morphology. They have very whitish hairs. They're often felty and fuzzy. But there's a lot of variation too within this group. And when we look at the principal component analysis again, we see that that blue lineage, B3, is falling in this intermediate morphological space. So it's not necessarily morphologically distinct based on leaf shape. And then we look at the ecological niche model. You can see that this lineage is limited in its distribution to Costa Rica and Panama, and it likes very wet habitats. So very wet secondary forests, and isothermality means that the variability within the temperature per month is less variable than it is throughout the year. So that tells us maybe that it likes more stable climates. So with that information, I can say that we can satisfy the ecological species concept and the phylogenetic species concept. And if we were to describe this species, given the breadth of knowledge that we have, and we thought about trichome morphology as well, I would move it into the center of these Venn diagrams again. And lastly, this B5 lineage that we recovered in the ETS data set is also pretty interesting. So let's learn a little bit about that one. These are the representative morphologies of this group. And it's pretty interesting because it has stridulose trichomes, which means they're oppressed and they're typically kind of condensed at the vein. And the veins of this lineage are also pretty elevated in the specimens that I've looked at. So that might be morphological characters that are distinct to this lineage, but not sure yet. And you probably noticed that this lineage is not included in the NOVA box plots, and that's because our sample size is 4. And for the NOVA analyses, we're looking at 5 different characters, so it couldn't be included. And this lineage also has an interesting distribution. It's distributed across the Sierra del Madre and also kind of at the intersection of the Trans-Mexican Volcanic Belt. So this is geologically a very interesting place. And this is also a higher montane lineage as well. And we can see that precipitation seasonality is especially important for this group. So based on that information, we can move B5 over here in the Venn diagram as it satisfies the phylogenetic and the ecological species concepts. And again, if we're looking at the breadth of the knowledge that we have about this lineage, we might be able to say that it moves to the middle of this Venn diagram, satisfying all 3 species concepts. So in conclusion, this is where we now lie based on what we know about these lineages. And again, while some of the data doesn't support that we're moving all these to satisfy all these concepts, when we're thinking about we have all these species or these clades that we've identified, how are we going to describe these species? When we think about, again, the breadth of knowledge, we need to consider that as we describe species. So in conclusion, we found that there are 6 species within Trema micrantha group B, the group that I've been talking about throughout the talk. And when we look at all the lineages in Trema micrantha and include A, C, and D, then we can say that there's 9 species total within Trema micrantha. So we noted that this is likely not a well-described species, and I think this evidence definitely supports that, because the diversity in this group has been vastly underestimated until now. And one thing that I've learned throughout this endeavor is that one line of evidence is almost certainly, in most cases, in at least some cases, is not enough. One line of evidence is not enough to describe species. When we're thinking about species, we need to think about the biological interactions that they're having, the environmental interactions that they're having as well, and look at their morphology. How would we diagnose these in the field? And also, most importantly, their evolutionary relationships. And with this information, now that we have a better understanding of the evolutionary history of this group, we can contribute that to, or we can then begin to look at diversification patterns of this group, and that helps us understand overall diversification patterns in the neotropics. And this is important because if you're not aware, in the neotropics, there's tons of diversity. 37% of floral diversity is concentrated in the neotropics. And with this more comprehensive understanding of the taxonomy of Trema, then we can begin to explore new questions. Someone else can explore new questions. So I think this is a great group to work on because there's just so much to explore. And throughout my talk, you've probably noticed I've shown a lot of herbarium specimens. And this work literally would not have been possible without the contributions of natural history collections. So all the collectors that have spent years in the field and years curating those collections, without them, I couldn't have even tried to take on a project like this. So I definitely appreciate their contributions more than I could describe. And big thanks to Morgan Kriebler, who's an undergrad in plant biology, and she helped me measure several Trema specimens over the course of a couple years. Thank you to new big lab mates, especially Marissa and Dr. Janice Valencia for all their help and friendship and support. Special shout out to my advisors, Dr. Garwood and Dr. Newbig, who I believe are some of the greatest advisors in all of academia, but they don't think that, but they are. And thank you to Dr. Weber for being on my committee, and I look forward to the questions that you might ask me pretty soon. Thank you to everyone who's serving in administrative roles in the plant biology program. Thank you to my mom. She's gonna cry, so I'm gonna move on. And I got some various funding throughout this project as an undergrad and graduate student. So it's been a real pleasure. All right, and now I'll take any questions. Thank you very much.